buddy. Okay. So, got a lot to do this weekend. Things have been stacking up, which is pretty typical. Sorry about the sound. My laptop's charging. And it's probably gonna overheat. Let me see if I can test. How well can y'all hear me on the desktop stream? Okay. So physiology and histology. Again. And this was mostly talking about the structure of the nervous system. And so spinal cord funiculuses or funiculi are the spaces in between the gray matter. I don't know if I think it's white matter maybe. There's a dorsal, a lateral, and ventral funiculus on every spinal cord. And they're paired. Yeah. Wait a minute, that's at the very bottom for some reason. Goodness. And structural. Hey, cheeky blocks. So, astrocytes, talking about astrocytes. They are star-shaped. That's why they're called astrocytes. We're just talking about the structure of these again. Thank you, cheeky blocks. I appreciate that. Neurons store 5 to 10 minutes worth of glycogen just in case the brain runs out of glucose. I guess that's why. That is what I guess. My professor said that glucose is more efficient than ketones, but that's actually incorrect. I looked it up, and ketones are more efficient than glucose. Especially for the, like if you're running on ketones, or ketosis is what I was going to say, but if you're running on ketones, and your brain is running on ketones, then there's just something different that happens. Your brain, you're able to focus better, I suppose. But your brain can utilize many sources of energy, lactate, as well as glucose. Your brain runs on Adderall and Red Bull. All right. That's pretty crazy. But anyway, nervous system. When your cells get damaged, they repair themselves. Question, how many times have you been thrown onto the court during a WNBA game? 0. 0 times. I've never even been onto an NBA game. Wait, what did I say? I've never been to an NBA game. That's what I meant to say. Astrocytes are important for the blood-brain barrier as well. The blood-brain barrier is created by tight endothelial junctions of the capillaries. These don't have any fenestrations. They don't have any clefts. And the basement membrane is continuous. But it also has high electrical resistance. It's very charged. P-glycoprotein. Yeah, there's something called P-glycoprotein, which is like, well, it says here that's an efflux pump in the capillary endothelium. Transport of chemicals and drugs out of endothelial cells back in the capillary. It helps with moving these chemicals in your cells out of the cell. That's what P-glycoprotein is for. My EBT got cut, bro. Sorry to hear that. Since yesterday was Halloween, I yelled to my wife, "It's a werewolf." When she turned, I showed her my full moon. That's crazy. That's crazy, bro. Are you a real doctor or just YouTube? I am not a doctor yet. Maybe, sorry if the channel name's misleading, but I will be a doctor in, about 3.5 years now. It's about 3.75 years, I guess. I am going to be a veterinarian, animal doctor. Hey, Fraudio Canopoima. I hope I said your name right. Ronald, thank you for the like. Rise and fire. On a serious note, my doctor said they should put Adderall in our water system because it would be a better place. Was he serious? I hope not. That's crazy. Putting drugs into the water supply is probably not a good idea. I'm not a doctor, but I can mallet your knee to check reflexes. Yeah, that's a pretty common reflex test. It really works. I don't know exactly the importance of it though, but yeah, I know what you mean, man. How many years does it take to become a veterinarian? Well, so if you were to just go through college with no special programs, 8 years. It'd take 8 years, at least at OSU, Oklahoma State University. That's where I'm at. But if you do things like the 3+1 program or the early admission program, it could actually take longer than 8 years normally. It might take 9 to 10 years because you have to get into vet school as well. You have to apply, and then they have to accept you before you're actually in. But hello, Faris. What are my origins? Tulsa. I was born in Tulsa, Oklahoma. But it only took me 3 years in undergrad, and vet school is going to take 4 years. No, I wasn't born in France. I was born in Tulsa, Oklahoma. You speak French? Oui. Yes. That's about all the French I know. I know some Latin, so maybe it. Oui means yes. Funkai, I don't know if that's how you say that, means French. Le means the. I know a little bit, but I don't know parler. Parler Coleman. I don't know. Is my concentration on a certain animal type, or do I know about every animal species? Well, so far, I don't know a whole lot. Now, Well, I do know a little bit. I learned a lot from undergrad. I know a lot about nutrition. The concentration, though, so far is just every animal species, I guess. I know a little bit about a lot, but I don't know a lot about a little yet. So, cattle, horses, um, dogs, cats. Yeah. Hey, Elizabeth. How old am I? I'm 21. There is a little bit of French that I do know from this one Roblox game I used to play called no, I'm 21. I don't know how to say that in French, but I know en avant, which means advance or something like that. En avant. And then there's hmm. Oh man, I can't remember the. Anyway, it was. Yeah, it was a Roblox game, but it was fun. Anyway, I gotta get back to this. What's my take on animal testing and research? Well, I don't know. That's a good question. Fares, you were born and you grew up in France. That's really cool. I've heard it's really pretty, but I've heard that a lot of main cities like Paris, or in the case of England, London, sorry, are kind of filthy. Is that true? I don't know. What's my take on animal testing research? So lab mice are raised in labs their whole life. So I don't know. Whether it's ethical or not. It's kind of depends on the opinion of the beholder, but I don't know. I don't want to be a researcher, so I just kind of try to stay away from all that stuff. I don't know. I don't necessarily like it, but at the same time, I'm not really involved in it, so I don't know. So there's plenty of chemoreceptors like dopamine, histamine, serotonin, scopolamine, which is a muscarinic, which is cholinergic. These are all things that the brain recognizes. Astrocytes also regulate ion concentration in the extracellular space. Astrocytes express potassium channels at a high density. They rapidly clear the excess accumulation of potassium in the extracellular space as well. Next, we're going to talk about oligodendrocytes. These wrap around neurons, which create myelin sheaths in white matter. Each oligodendrocyte wraps around many axonal processes. This insulates neurons, which makes the signal faster via saltatory conduction and nodes of Ranvier. But Schwann cells also make myelin, except they make them for peripheral nerve cells in the PNS, peripheral nervous system. They make a single myelin segment per peripheral nerve. Then there's microglia. We've already talked about this before, I know, but these are the macrophages of the brain. Um, hey, Giselle Scott, how you doing? Then there's the cerebrospinal fluid. Do I have to take organic chemistry? I assume you mean to get into vet school. Yes, you do. I did. I thought it was fun, but it was challenging, for sure. I'm a weird guy. I like chemistry. A lot of my classmates do not, but I understand. Um, there's a lot of classes I don't like that I had to take. I used to be your doctor? Really? I find that hard to believe, considering I'm not a doctor yet. But, alright. Do I look like your doctor? Is that what you mean? Cerebrospinal fluid surrounds and permeates the central nervous system. It functions basically as a cushion. It's a shock absorber, right? It serves a chemical sink function, if that makes sense. What that means is it exchanges materials with the brain, interstitial fluid. There's nutrients flowing around. And it makes sure that the brain's always floating, for lack of a better term, in nutrients. It is made by the choroid plexus. Cerebrospinal fluid is made by the choroid plexus, which is present in the lateral, third, and fourth ventricles. The choroid epithelium, which is ependymal cells, and then found with tight junctions. Can you treat a gunshot wound on a human as a veterinarian? Yeah, you could. I mean, veterinary medicine is very, very similar to human medicine. It only differs, I almost said diffles. It only differs in the fact that animals have... Yes, Giselle, yes, I get it. It only differs in the fact that, okay, for example, dogs have 6 lobes in their liver. Humans have 4 lobes in their liver. But they still have a liver. Dogs still have a liver. Cats have a liver. Humans have a liver. It still serves the same function. There's very many differences, tiny differences, but overall everything's the same. Suturing technique. So yes, a gunshot wound. Thank you, Giselle, by the way. Thank you for the subscription. A gunshot wound, as long as you get some forceps, remove the bullet if it's superficial enough, I guess. I mean, if it's too deep, then don't want to maybe risk it, but yeah, you can easily suture up a wound on a human. It's the same exact technique, I assume. I don't really know, but I mean, suturing is suturing. So as long as the wound gets closed and heals naturally, I think it's okay. If you're going to use anesthesia, you might need to do some calculations. Be careful. Or you just might need to forego anesthesia, and it would be painful. But in a situation like that, gotta do what you gotta do, right? But you still, as a veterinarian, you still learn a lot about medications. You learn a lot about anatomy. You basically, you know all the same things that a human doctor knows, just specialized. You know it about, or you know it relating to the dog or the cat. Cerebrospinal fluid also provides buoyancy, and apparently it reduces weight, or it reduces the weight of the brain by a factor of 32 times because of that buoyancy. It can buffer ionic or metabolic concentration changes and also pH changes. It's a plasma filtrate, basically. The choroid plexus is a part of the brain. I think it's under the ependymal cells. But cerebrospinal fluid fills ventricles, circulates through the channels that link the ventricles, goes into subarachnoid space, goes down the spinal canal, around the brain, and then it's absorbed by subarachnoid villi into the sinus. And it's replaced 3 to 5 times a day. So it's replaced fairly often by the body. Your body's basically always producing cerebrospinal fluid. Okay. Cerebrospinal fluid's absorbed in the arachnoid villi, or by the arachnoid villi, into venous sinuses, veins, and lymphatics. And it's absorbed from the subarachnoid space by pressure through one-way valves. But the failure of this CSF to be absorbed adequately results in ventricular expansion in the brain, which can be painful. That's what is seen in hydrocephalus, especially if the animal is young, very young, puppy, right, or a kitten. When their skull is still malleable, still able to expand, hydrocephalus is really not good, because then it enlarges their skull, makes it look like this. Okay, and then there's ways to collect it for histology or whatever. A spinal tap is what that's called, I'm pretty sure. But the cerebrospinal fluid is supposed to be clear and colorless. Not a lot of protein. But it can change color depending on diseases, etc. For example, a horse with herpes virus myeloencephalitis would have cloudy yellow cerebrospinal fluid. Why is it yellow? Because it's full of albumin, which is a certain kind of protein. Okay. Then, how the nervous system's organized. Obviously, there's the central, there's the peripheral. Then there's many divisions. There's the telencephalon, diencephalon, cerebellum, brainstem, and spinal cord. The diencephalon contains the thalamus, the subthalamus, and the hypothalamus. The brainstem contains the midbrain, the pons, and the medulla. The telencephalon is composed of the cerebrum and a large mass of connecting pathways. Some of these connecting pathways are corpus callosum, basal ganglia, which are actually basal nuclei. They're nuclei. They're not ganglia. But That was the old name for it, basal ganglia. All right. The cortex in the forebrain has the frontal lobe, which is for motor, or is part of the motor cortex. The parietal lobe, which is sensory, touch, pain, proprioception. The occipital lobe, which is for vision, and then the temporal lobe, which is for hearing and proprioception. And if you were to remove any of these, you would see a decrease in functioning regarding what they govern. Does daylight savings time harm animals? No, not really, because daylight savings time is a human construct, and daylight savings time, all it really does is it doesn't change time. First of all, time never changes. Time's always going forward. But what it does do is change our what's the word? Our attempt to govern time. All it changes is our clocks. It doesn't affect how time actually works. Some clinical signs of cerebral disease are seizures, narcolepsy, altered consciousness, blindness, behavioral changes. If you see any of these, could be a sign of cerebral disease. I'm a poet and I didn't know it. Nervous tissue can only respond in a certain limited number of ways. Once the tissue's damaged, it can only heal itself in a certain way, or respond in a certain way, like seizure. That's when the electrical discharge of neurons is excessive and uncoordinated. You just start moving around weird, right? It could involve other regions like vision, sound, smells, panic. For example, if you get a certain pattern of vision, you might have a seizure, epilepsy, flashing lights, right? Certain sounds, certain smells, a panicked state. All these things can cause seizures. Because all a seizure is your brain starts discharging electricity in a certain way that causes you to have a fit, basically, have a convulsion. Or it could be due to a head injury, could be developmental, could be due to a tumor, could be due to a toxin, hypoxia, infections, such and such. Could be due to a cholesterol granuloma, which is a benign thing that develops in the choroid plexus, in this case a horse, but it causes seizures. There's ways to evaluate seizures: CT scans, MRIs, electroencephalography. Or you can just say, oh, this person's or this animal's like convulsing. It's a seizure. That's an evaluation. You say, oh, they're having a seizure. They're having a seizure. Okay. Yeah, toxins can cause seizures. Cerebellum. The cerebellum is 10% of the brain by volume, but it's 50% of the brain in terms of neuron numbers, at least in humans. It receives input from almost every type of receptor in the nervous system. There's 3 divisions of it. The vestibulocerebellum, which maintains head and body position. The spinocerebellum, which regulates muscle tone. And then the cerebrocerebellum, which coordinates motor movement. The cerebellum doesn't initiate movement, but it takes signals from the things initiating movement and then coordinates those. It helps with rate, range, and direction of movement. Many diseases that can affect the cerebellum's function. Okay, then there's the diencephalon. We talked about this a little bit. Composed of 3 regions. The thalamus, which is the integrating station for sensory information going to the cerebral cortex. Then there's the subthalamus, which is the integrating station for motor function from the basal ganglia. Then there's the hypothalamus, which it deals with connections to the autonomic nuclei in the brainstem, and it is part of the endocrine system. The brain stem is composed of 3 structures: the midbrain, pons, and medulla. The midbrain is composed of the origins of motor neurons that control eye movement. It relays signals that relate to hearing and vision as well. The pons is composed of somatic motor. Sorry about the connection. Looks like connection shut off for some reason. But we're back on here. And looks like we're back on the desktop screen. Okay. Where was I? And the medulla, which is composed of somatic motor nerves to muscles of the neck and tongue. It's involved in control of blood pressure, heart rate, respiration, and digestion. And then the reticular activating system, it's also involved with. The way this differs from human medicine in veterinary medicine is actually it's very, very, very similar. But the brainstem centers coalesce and organize into columns which go down the entirety of the spinal cord. There's dorsal and ventral branches. The dorsal branches are sensory and afferent, while the ventral branches are motor and afferent. Efferent, that should be. Motor, efferent. Okay. Whoops. Okay, the spinal cord, it's continuous with the medulla and extends the length of the spinal column. It consists of segments, and each segment has a sensory afferent nerve and a motor efferent nerve. And they all connect to specific regions of the surface of the body, which are called dermatomes. Neurons, yep. Okay, we're done. That's it. That's it for that. What else? Oh yeah, immunology, epidemiology. Oh, I want to do this. Whatever. Okay. I actually, I don't. I've got a quiz to take. I'm gonna take that quiz. Epidemiology. And then I'll probably be back later making another stream. But anyway, thanks for watching, y'all. I'll see you in the next one as per usual. So, bye-bye.